Hello there. So I'm Aaron Petkau, and I work with the Public Health Agency of Canada over in Winnipeg at our large microbiology lab over there. And we've been using Galaxy for quite a number of years, both as a method of performing high throughput research, but also we've been developing software that sort of sits on top of Galaxy and assists us in a few more automated ways that I'm going to describe right now with our project IRIDA and its pipeline in Galaxy called SNVPhyl. Okay, so at our lab, well, we deal with quite a lot of different sorts of data, but the one particular area that I'm involved in a bit is for outbreak detection and response and investigation. So in particular, we're focused on bacterial pathogens, in particular foodborne bacterial pathogens. So we get a lot of samples from a variety of sources, such as food, environmental, so farms or processing plants, or clinical samples, and we're interested in any sources of contamination on those different sources of food or environmental samples. So what we do is we sample and culture the bacteria that we get in, and we're interested in generating these fingerprints for these different bacteria. And once we have a set of fingerprints and a large database, we can then start comparing them to determine whether or not, for example, one contaminated food was the actual source of some illness that different people have suffered from, or start excluding maybe some farm from being the source of some particular illness. And what we're trying to do is move towards using genome sequencing, in particular whole genome sequencing, to generate these genomic fingerprints. So we are able then to get right down to the individual nucleotide level, which gives us a lot higher resolution picture, but also gives us access to a lot more data for further analysis, such as, for example, looking for antimicrobial resistance genes. So this is where our project IRIDA started from, Integrated Rapid Infectious Disease Analysis. What we want to do, or what we set out to accomplish with this project, is to develop a system for managing, in particular, whole genome sequence data and sample metadata. And we also want to be able to analyze that data and generate reports. But we also wanted to have the capability of sharing these sequence data, for example, between maybe different instances of IRIDA across different labs, or being able to link up to external tools for more in-depth data analysis that's not really able to be provided by some of the built-in tools. So the overall architecture of this IRIDA project, the main component of it right here is a web application written in Java, which has two main entry points: a web interface for people to interact with, as well as a REST-like API. And this allows people, for example, to upload sequencing data from the web interface and organize it into different projects or different folders, but also allows more automated systems like a genome sequencer to upload data in bulk without any human intervention. And one difference, or one thing I'd like to highlight over for anyone who's previously seen our project, is that we've gotten all of our code up on GitHub now, so it's a completely free and open source project that's available for anyone else to download and install. So once you've got data into our system, the large files are managed on a central file storage area, and data analysis is handled by a compute cluster, of which we're using Galaxy to actually handle running workflows and tools and generating the results for us and storing some of the provenance information. In addition, though, while we have a few certain specific built-in workflows into IRIDA, we also allow people to export data outside of IRIDA into different environments for more custom analysis, such as into a command line-based environment where we generate a series of links so you don't have to re-download all your FASTQ files, or into a custom Galaxy instance where you can then run whatever tools you want in any standard Galaxy installation. So getting data into IRIDA, we've written well, the first way you can do that is just uploading FASTQ files, but that's pretty tedious for lots of files. So we have this uploader tool written in Python, and we're currently using it by running on our genome sequencers right after we finished a sequencing run, and this will communicate with our IRIDA application and essentially look through the directory structure produced by an Illumina MySeq instrument and upload any of the FASTQ files in there and organize them into the different projects that were defined beforehand by a user. Getting data out of IRIDA, we have three main ways. You can directly download all of your FASTQ files, but we're trying to discourage that as it leads to data duplication of these large multi-gigabyte files. So we provide a command line linker program which will interact with IRIDA over its API again to get the full file paths on our storage area and generate a series of soft links and directory structures so that you can more easily then run command line tools on your sequence reads. But in addition, we also have this built-in tool in Galaxy which will interact both with Galaxy and with IRIDA via an API. So you'd start off using it by going into Galaxy, and in the left panel in your import data or get data section, you're able to click on an IRIDA importer. And this will bring you to an IRIDA instance where you can then go and select what samples you want to import. And then you can click on the Send to Galaxy button right here, and it will go and run a tool in Galaxy to construct dataset libraries for you, link up your data, and then construct a dataset collection with all of your files in a user's history. Analysis is handled by, again, Galaxy that's sitting over top of a compute cluster. So what we do with Galaxy is we have a number of predefined workflows that we're managing within our code base. And from IRIDA, a user is able to go and quickly select what samples they want to run workflows on. From this set of samples, they can go and select from a number of pre-built pipelines. And once this is all configured, all the information, such as the parameters they may override or the set of samples, are sent to Galaxy over the API. And workflow is triggered, and IRIDA will monitor that workflow until completion, at which point it will store some of the most important files and then provide you with custom visualizations and reports directly in our IRIDA system. So one of the particular pipelines we have in IRIDA is our SNVPhyl pipeline for generating whole genome phylogenies from single nucleotide variants identified from bacterial genomes. So in short, what it does is it takes a reads and a reference genome. In this case, our reference would be a bacterial genome, often either some standard reference genome or something that's been assembled from the set of genomes you're interested in. And you would identify the variants. We have a lot of customized filtering involved in this pipeline to remove variants in repetitive regions or variants that could potentially be problematic. And once you've identified your set of high-quality variants, you can generate a phylogenetic tree. And this is the main method we're using to sort of generate the genomic fingerprints and determine whether or not different bacterial samples are match or non-match for either outbreak investigation purposes or for other more research purposes. And all of our code is on snvphyl.readthedocs.io. It's implemented as a Galaxy workflow, so you can go and install the tools. It uses a lot of standard tools, but we have a few custom scripts for filtering and sort of merging all of our data together. In addition, we've also developed a command line interface around our SNVPhyl pipeline. The idea here is I wanted to more support batch execution or integration with other systems. So, this allows you, it's a Python script where you can pass in a directory of your FASTQ files, a reference genome, override a couple of key parameters, and then you can pass in either your Galaxy credentials and have it automatically upload all your files to a Galaxy instance, prepare dataset collections and execute a workflow and download results at the end. Or you can also pass an option to deploy a Docker container with our tools and a Dockerized version of Galaxy. So this bottom one allows you to quickly get up and running with our pipeline for anyone who wants to try it out. In addition, our IRIDA application interacts with external services. So this is done via a number of APIs. IRIDA itself has an API where you can get the different sequence reads or analysis result files. And a few different services we interact with or we've implemented is our NCBI Sequence Read Archive Uploader tool. So here, especially used for research purposes where you want to submit your data to NCBI Sequence Read Archive, we have a form you can fill out where you can enter in some of the required metadata and it will automatically go and upload all of your files in batch to NCBI. We also interact with some other tools. Genghis is a desktop application for integrating geographic and phylogenetic information. So there's a plugin in Genghis that was written which will interact with IRIDA over our API to download a phylogenetic tree and geographic information for any samples you have and be able to display it in a few different customized views that have been in development. And there's a number of other tools we plan to integrate into or along with SNVPhyl. One is a sister Salmonella in silico typing resource, which allows you to upload a Salmonella genome and have it be compared to a large and growing database of Salmonella genomes for serotyping or for a custom core genome MLST typing. We also plan to integrate CARD for identifying antimicrobial resistance genes, as well as Island Viewer for genomic islands. Now, I wanted to get a bit more into how we're actually using IRIDA at our lab in the Public Health Agency. So we actually have, our overall infrastructure is, we currently have a 1,500 compute core cluster and 400 terabytes of storage, though we're continually growing. And on top of that, we have two different types of Galaxy we're using. We have one that's solely dedicated to running IRIDA workflows. So this is not really meant to be logged in by a human except for debugging purposes. And it's just interacted with over an API with our IRIDA application. But we also have another research-oriented Galaxy, which is kept a bit more up to date with the latest Galaxy. And this one is where all of our users are able to log into and run their own custom pipelines. And both of these submit to the same compute cluster. And in addition, though, we also use Docker Galaxy. Over the last year, we've actually gotten or integrated Docker Galaxy into our code base for integration testing. But we also have a Dockerized version of all of our tools and Galaxy available for those who want to sort of quickly start up a Galaxy-based compute engine for IRIDA. Data usage over the years for IRIDA, again, we started this a number of years ago, back in, say, well, before 2013, but we deployed our initial IRIDA instance at our lab in 2013. At that time is only really for managing our sequence data and some other metadata. And then we later on linked it up with Galaxy for executing workflows. But over the years, we've grown quite a lot in terms of the amount of data we've managed to store. And we're often, the rate at which we're growing is increasing all the time as we add more and more sequencers to our lab and are running them more often. Right now we're up to about 18,000 samples and about 14 terabytes of sequence reads. This has all led to a large increase in the number of Galaxy jobs we're running. Again, we have multiple Galaxy instances at our sites, and because we're running such large workflows on many different bacterial genomes, for example, hundreds of genomes, and each of those workflows may run themselves thousands of jobs before a result is produced. We've got a huge increase in the number of Galaxy jobs we're trying to manage, up to about 1 million right now. So for ways that we're trying to more optimize our cluster resource usage, you can check out poster 4 on a tool called on dynamic tool destinations, where we're trying to associate resources that are required by a tool with the actual input file sizes. For the future, we're actually currently right now working on distributing MySeq instruments plus IRIDA across Canada. The idea being that some will be hosted in Winnipeg in our main lab, where we have a lot of compute infrastructure, but other instances can be hosted at other labs and linked up through our sharing network. And we want to modernize labs and enhance our epidemiological investigations with sequencing data. And more information, I've given a lot of URLs here, but really the main one is our irida.ca site, from which you can find most of our other projects. But we also have our SNVPhyl program on Read the Docs, and we have all our code on GitHub. And we have 2 posters that you can check out right afterwards, 4 and 14. So there's been quite a number of people involved in this project over the years. There's been lots of work on it, but along with all of these people, I'd also just like to thank the Galaxy team, because they've done quite a lot in terms of adding in some of the features that we use quite extensively, such as data set collections and better ways of developing tools. So that's it, and does anyone have any questions? Yeah, that's part of IRIDA. That's something in IRIDA, not in Galaxy. Yeah. In Galaxy, you mean? Possibly feasible. It's not something that we worked on because we have it in IRIDA, but I believe how it works is you define your credentials for NCBI in a config file, and it goes and uploads some data. We've had some issues kind of getting it up and running as they change their API. So I would say it would be feasible, but maybe a bit more work to develop some kind of tool like that.
So I was also wondering, you mentioned the fingerprints for the various bacteria. Where did those sequences come from?
Where did the sequences come from for, sorry, could you repeat that?
Yeah, like what are you looking for when you're making fingerprints? Is it 16S or?
Oh no, it's whole genome. We're looking at SNPs or SNVs, variants across the entire genome.
Okay.
Yeah, so that's one method. Another method is looking at genes, variants in genes across the entire genome.
Gotcha.
Yeah.
Cool. All right, let's thank the speaker one more time.
Thank you.